Adaptation to Environmental Change, which is co-organised by Stu Plaistow and I. First, before we begin the talks, what is the motivation for this session? Well, we already know that organisms can adapt to environmental change. We have a rich history of examples of this, including some classic cases from Liverpool here. Despite this, we don't fully understand how organisms manage to adapt. How are the rates and limits to adaptation actually determined? Here are some of the outstanding problems. First, despite a wealth of next-generation sequencing data, we still understand the connection between DNA sequences, phenotypes, and fitness only poorly. New statistical methods are needed to illuminate the adaptive process. It is also becoming clear that understanding DNA sequences and their evolution alone may be insufficient. For instance, how do organisms remain robust in the face of adaptation? In addition, phenotypic plasticity and non-genetic inheritance can each contribute to phenotypes and adaptation. The effects of these processes also need to be incorporated. Conveniently, I'm really delighted to be able to introduce a group of speakers who will cover many of these aspects and who will give us a taste of the cutting edge of our study of adaptation. So just a brief warning, 2 of the talks within this session are actually 30 minutes long. Those talks start at 15:45 and 16:15. Well, I'm really looking forward to these talks, and I hope that you enjoy them too. So first up, we have Gabriel Yvon de Rocha from the University of Exeter, and he will be talking on metabolic traits that underpin thermal adaptation in phytoplankton.
Great, so thanks very much for the introduction, Raj, and thanks a lot, Stu, for inviting me along to have the opportunity to give this talk. I'm going to talk about some work that's been carried out by my PhD student, Dan Padfield, and my postdoc, Eliza Sharm, with the general idea to really try to understand the mechanisms that underpin the evolutionary responses of phytoplankton to warming. And I guess the backdrop for a lot of this work comes from evidence from process-based biogeochemistry models, which predict general declines in marine net primary production over the 21st century, principally driven by enhanced grazing rates and enhanced nutrient limitation at higher temperatures, which exceed the kind of stimulatory effects of temperature on phytoplankton growth. However, none of these ecosystem models are actually able to account for the effects of rapid evolution, the capacity for these photoautotrophic microbes to rapidly adapt to changes in the environment. And this is because we understand relatively little of the fundamental mechanisms that actually underpin the evolutionary responses of phytoplankton to warming. So this is a toy diagram of what we might expect to see if a phytoplankton population is able to rapidly adapt to an increase in temperature. So this is a reaction norm of the thermal tolerance for fitness or for population growth, a component of fitness. And if we imagine this cooler adapted population, they have an exponential increase in growth rate up to an optimum followed by a rapid decline. And in their current environmental condition, they have a growth rate here given by the blue dot, which is close to the optimum temperature, so they have a bit of a buffer in the face of environmental warming. But if environmental warming occurs to such an extent that it pushes the temperature beyond that optimum, then the growth rate declines rapidly, and we end up in this dangerous situation here where we have severely depressed growth rates, and the population may be on the edge of extinction. If the population is then able to rapidly adapt to that change in temperature, i.e. it's able to right shift that thermal tolerance curve, increasing the optimum temperature, the net effect then is to increase the population growth rate in that new environmental condition, and we have an evolutionary rescue, let's say, of that population. So what we want to understand is what mechanisms might facilitate these right shifts in the thermal tolerance curves for population growth in phytoplankton. So phytoplankton growth is underpinned by 2 fundamental physiological pathways: photosynthetic carbon fixation and respiration. And ultimately, the balance between photosynthetic carbon fixation and respiration determines the amount of carbon that the organisms have available then to allocate to growth. But the thermal response curve, or the thermal sensitivity of respiration, is often more sensitive to temperature than that of photosynthesis, which means that the balance between respiration and photosynthesis, or the carbon use efficiency, which is 1 minus the respiration to photosynthesis ratio, tends to decline as temperature increases, because respiration is more sensitive to temperature than photosynthesis. And that's given by this black line here. Photosynthesis is in green and respiration is in red. Now imagine that there is some threshold carbon use efficiency below which the phytoplankton populations can no longer allocate sufficient carbon to provisioning of growth. If the carbon use efficiency falls below that value, then growth rate is going to decline as a function of increasing temperature. So imagine this scenario here where we have the blue population and we increase the temperature to the red point here. The carbon use efficiency now is below that threshold value, and the population can no longer allocate sufficient carbon to growth. So how might evolutionary adaptation adjust the respiratory and photosynthetic physiology in order to enable elevated population growth and the evolution of elevated thermal tolerance? Well, one possible scenario is given by this red dashed line here. If the cells are able to down-regulate their per capita respiration rates, then the net consequence of that is then to increase the carbon use efficiency at that higher temperature to around about the threshold value, and thereby enabling the populations to grow well again at that higher temperature. So we explored some of these hypotheses using a tool called experimental evolution, and the first experiment that we carried out was on a population of freshwater green algae called Chlorella vulgaris. The long-term kind of ancestral laboratory temperature that the organism had been cultured in was at 20 degrees. We took that population at 20 degrees and then we divided it amongst 5 treatments of increasing temperature. We maintained the populations in semi-continuous batch culture for around about 100 generations. And then after 10 generations and at 100 generations, Dan Padfield carried out a whole bunch of different metabolic and phenotypic assays to try to understand the mechanisms of adaptation to these different temperatures. So these are the population growth rate data as a function of the selection temperature after 10 generations in those different environmental conditions. And we can see that population growth increased as an exponential function of increasing temperature up until 30 degrees. And at 33 degrees, the populations were constrained. The population growth was no longer where we would expect it to be on the basis of the exponential relationship between temperature and growth rate. However, after 100 generations of selection at that elevated temperature regime, given by the red points, we see a significant increase in population growth rate. So it looks like these organisms have evolved tolerance to that higher temperature. They've been enabled to increase their growth rate at those higher temperatures. So what mechanisms might have facilitated this adaptive response at 33 degrees? So these are the data from the thermal response curves for photosynthesis and respiration. Photosynthesis is in the left-hand column, respiration is in the right-hand column. The data after 10 generations over a kind of acclimation or a plasticity timescale are in the top panels, and the longer-term data after 100 generations are in the bottom panels. The key point that I want to draw your attention to here is that the two treatments are in blue and in red, and we see very little difference between the warm populations and the control populations after 10 generations. But after 100 generations, we see substantial differences between the 20-degree populations and the 33-degree populations. And the main effect is on respiration. We see a significant downregulation in the per capita respiration rates of those populations at 33 degrees. The consequence of that downregulation in respiration was to then increase our carbon use efficiency. So we've got carbon use efficiency on the y-axis here and selection temperature on the x-axis. The evolved lineages after 100 generations are in red, and the 10 generation data are in black. And you can see that at the higher temperatures, there's a significant difference between the 10 generation data and the 100 generation data. So we get a significant increase in the carbon use efficiency, the fraction of photosynthetic carbon that can be allocated to growth, driven by that downregulation in the respiration rates. Okay, so the next experiment I'm going to talk about is work carried out by Eliza Sham, who's been doing her postdoc with me. And we wanted to see whether these mechanisms of thermal adaptation are general? Do they apply to different phylogenetic groups of phytoplankton? And perhaps more importantly, more ecologically relevant marine phytoplankton. So we used Thalassioceras pseudonana as a model organism. Eliza established a clonal founder population and then distributed that population across 5 different experimental treatments. So the control long-term ancestral temperature was 22 degrees, and we then looked at moderate warming and at extreme warming, and then at 2 different levels of fluctuation between the control and the high degree of warming. And I'm just going to talk to you about these results at the stable temperature. Eliza spoke this morning about the responses to fluctuating temperature. And then after 100 and 300 generations, Eliza made a whole bunch of phenotypic and metabolic measurements. Okay, so this is a graph of the fitness trajectories over the time course of the experiment. We've got population growth on the y-axis and time of the experiment on the x-axis. And you can see that for each of the treatments, we have a kind of positive trajectory of population growth. So even in the control, we're getting increases in fitness over time. So these organisms are adapting to our laboratory culture regime. But at 26 degrees, we see the most marked or the most rapid increase in fitness over time. And then at 32 degrees, the extreme warming treatment, we see some quite interesting patterns. So initially, population growth is stimulated by the high temperature, and then it rapidly declines. And for about a year and a bit, the populations have a very, 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 very low growth rate. After about a year or 100 generations or so, we then see a rapid increase in population growth, which looks a little bit like a kind of classic evolutionary rescue type response. So these are the thermal reaction norms for population growth. So each experimental treatment was then taken out and then exposed to a broad gradient in temperature. Population growth again was measured at that broad gradient of temperature. The blue treatment here is the control, the 22-degree population. You can see that the optimum temperature is relatively low. And then at the 2 warmer treatments, at 26 and 32, we've got a significant increase in the optimum temperature for growth. And we can see, particularly in the 32-degree populations, that population growth, they're still doing okay at 40 degrees. So these organisms have changed their thermal tolerance curve in adapting to those different temperature regimes. These are the acute responses of gross photosynthesis and respiration to acute variation in temperature. And again, in much the same way as we saw for the green alga, we see downregulation relative to the control in both the photosynthesis and the respiration. And again, the effects are most marked for respiration. So the 32-degree population in red and the 26-degree population in green have got significantly downregulated per capita respiration rates relative to the control populations. And again, the net effect of that is to upregulate or to increase the efficiency through which carbon can be allocated to growth at those higher temperatures. So here on the y-axis, you've got the carbon use efficiency of the evolved lineages relative to the ancestor at the different assay temperatures. And if you look at the 300 generation data, you can see particularly for the 26-degree populations and 32-degree populations, substantially increased carbon use efficiencies at those higher temperatures. So again, for this organism, it looks like the evolution of elevated carbon use efficiency is facilitating the adaptation to those higher temperatures. Okay, so I just want to finish by talking about another experiment that Eliza did. So this is a photograph of our long-term warming experiment down at the Freshwater Biological Association in Dorset. We've got 20 experimental mesocosms that hold a cubic meter of water, and half of those mesocosms have been warmed up by 4 degrees, and the experiment's now been running for a decade. So what Eliza did here was she isolated Chlamydomonas reinhardtii from each of these ponds. So Chlamydomonas is cosmopolitanly distributed across the warmed and the ambient treatments in this system. She brought them back into the lab, and then she characterized a whole bunch of fitness and phenotypic trait characterizations. So this is a graph of the thermal tolerance curve for population growth for the warmed isolates in red and the ambient isolates in black. And the main effect here is there is a significant difference in the optimum temperature for population growth between the warmed and the ambient isolates. and the replicate-specific values are plotted in the box plots here. So there's about a 2-degree difference in the optimum temperature for population growth between the warmed and the ambient populations. She also characterized the responses of photosynthesis and respiration to temperature variation. So the left-hand graph is photosynthesis, the right-hand graph is respiration. And fascinatingly, what we're seeing in this experiment is that rather than down-regulating per capita respiration rates, the populations from the mesocosm seem to be up-regulating per capita photosynthetic rates, but with the same net effect. So the up-regulation of photosynthesis increases the carbon use efficiency, it increases net photosynthesis. So ELISA then cross-competed all of the warm adapted isolates and the ambient adapted isolates against one another at a relatively low temperature and at a high temperature. And also, we also have the kind of net photosynthesis estimates from those thermal response curves for respiration and photosynthesis. So this graph on the right-hand side shows you the competitive fitness of the warmed isolate as a function of the pairwise difference in net photosynthesis between those two isolates. And to cut a long story short, we see a positive correlation between those two variables. So it looks like competitive fitness is related to the differential in net photosynthesis. Okay, so just to conclude, we've seen that adaptive responses to warming are driven by increases in carbon use efficiency and net photosynthesis. These seem to be conserved across different phyla. And we also see similar mechanisms, but not identical mechanisms, occurring in more complex semi-natural experimental systems. So thanks very much for your time. Take any questions.
Right, we have time for one quick question. If you want to give that question, we need to use the microphones, though. Yes. Could you come to a microphone, please?
Hi, Gab. This isn't particularly better. I just wanted to check, are there other ecologically, either other adaptations which occur, so for example cell sizes or masses or other things which are happening at the same time as these thermal adaptations that might have ecological significance?
Right, so in the thermal tolerance curves for growth rate that you saw in the Chlamydomonas example, yeah, you see huge plasticity in cell size, but the plasticity is linear, and the slope of that reaction norm with temperature is identical between the warmed and the ambient isolates. So it doesn't look like there's any evolutionary effect on the plasticity of cell size between the warmed and the ambient isolates, but they're both equally plastic in the same direction, yeah. Which is intriguing. Yeah. Cheers.
Thank you, Gabriel. Our next talk is from Louis-Miguel Chauvin. He's from the Centre for Functional Ecology and Evolution at Montpellier.
Okay, so I'd like to thank the organizers also for inviting me to this session. So yeah, I'll be talking about the contribution of phenotypic plasticity to adaptation and population persistence in new environments. So this talk is essentially theoretical. So when I have longer talks, I put pictures of animals and plants, but I figured microalgae are at least as important, so I put nothing then, just theoretical graphs. So, well, just to define what we're talking about, phenotypic plasticity is just the phenotypic response of the phenotype of a given genotype to its environment of development or expression. So you can represent this as a reaction norm where you have an environment of development, and this is the expected phenotype of a given genotype. You can call this a breeding value if you're working in quantitative genetics. So, so then when you have something like this, you can either have measured two contrasted environments, and then difference between the phenotypic value expressed in these environments is what you would call plasticity. Or you can just try to look at the functional shape of this, and in this case, you can approximate this as a line, and then the slope measures plasticity, for instance. So the fact is that phenotypic plasticity has mostly been seen as a mechanism for coping with stationary environmental heterogeneity. But the problem is, like, when we're dealing with trends such as global warming, they can push environments beyond what were the previous ranges. And then organisms are exposed to what were previously rare or extreme environments. So, well, this is like, for instance, for global warming, you see this large fluctuations from year to year, but then you have this trend that goes beyond the usual, the previous mean and distribution. So then the question is, how does phenotypic plasticity interact with this with eco-evolutionary dynamic process in novel environments? And there's some accumulating evidence on this, mostly coming for instance, from meta-analysis of invasions, where it's an example of adapting to a new environment. And there's some evidence that you find higher plasticity in invasive species. So the most plastic species were more able to invade. There's also now accumulating evidence for evolution of plasticity in invasive species relative to their ancestor. But in terms of theory and concepts, this hasn't been addressed very much until lately. I mean, historically, there have been discussion of the Baldwin effect, which was sort of, so it goes back to the late 19th century, and it was later interpreted to mean that adaptive plasticity may allow population to persist when they arrive in a new environment, and this allows for evolution to proceed later, which is not what Baldwin meant, but this is how it was interpreted anyway. But there are a few quantitative arguments, and actually there's little consideration about the evolution of plasticity itself. So, for instance, if you think, without considering the evolution of plasticity, if you just think of constant plasticity, and then how this interacts with evolution. So consider a case of environment changing at constant speed, like this moving optimum. Then if the mean trade value was originally at the optimum, then when the optimum is moving, the mean trade value is moving farther away from the optimum, but if there's stabilizing selection, this selection will bring it back towards the optimum, and at one point you will reach an equilibrium between directional selection bringing you back towards the optimum and the optimum moving, and then you reach a stationary distance from the optimum, and this determines your lag load. For instance, if the mean trade value at equilibrium is outside of this gray region that allows positive growth rate, then the population goes extinct. So what happens if you have constant plasticity? You put this in the picture. Then in each generation, the mean trade value, so plasticity brings you back towards the optimum. So in effect, directional selection on the trait is lower. So this is the kind of argument about plasticity counters genetic adaptation. Of course, if you measure the trait value in a constant environment, in a common garden, you would see that it's responding less to selection over generations than if there were not plasticity. But what's happening in the wild is that you're not in a common environment. So there's also the plastic response, and the overall effect, if you have partially adaptive plasticity, is that you track the optimum better when you combine adaptive plasticity and genetic evolution. So, which sort of makes sense intuitively, but you can find a way to quantify this. So we did this in a model a few years ago, and from this you can define the critical rate of environmental change beyond which the population would go extinct. It depends on classical parameters that were in all these models, like the maximum growth rate of a population of a well-adapted phenotypes at the optimum, or it's reduced by the phenotypic variance in the population, which is implicit here. The critical rate of environmental change is larger when you have stronger stabilizing selection, because even though this will cause individuals to have lower fitness when they deviate from the optimum, it will also cause more rapid response, genetic response, and this is what dominates these effects. Well, there are other effects, but the main point here was that the critical rate of environmental change is larger when the plasticity, which is the slope of a reaction arm, which is assumed linear in this case, is closest to the slope of how the optimum changes with the environment. So we use this model to sort of argue that these are the types of parameters that you would need to measure, and are especially for this parameter of movements of the optimum are difficult to obtain and haven't been measured in that many cases, but it's starting to change now. But the point with this model is, like, we assumed there was some kind of partially adaptive plasticity that was going on, and obviously this cannot go on forever, just as the phenotype cannot evolve to infinity forever, even without plasticity. This is just a model to guide our intuition, but then people would argue that when populations, when under sustained environmental change, this population become exposed to environments that were previously rare or extreme. And then there's this argument going back to this Gallenborg paper from 10 years ago that where basically you have relaxed selection in those extreme environments in the past. So this means that in the environments that were common, the reaction norms should be tracking or shouldn't vary more, and then outside of the common environments, you will have these reaction norms going basically anyway, anywhere. So, but this was mostly a qualitative and verbal argument that assumed no genetic constraint on reaction norm shape. So we were trying to make this argument a little bit more explicit in a review and model paper that's going to be coming out in Field Trans next year. So if you consider the trait value expressed in an extreme environment, this responds both to direct response to selection in this extreme environment and also to indirect response to selection in the more common environments. And so if you have an environment that fluctuates and once in a while because extreme environments, you encounter them once in a while, it's just more rare. So then what happens is like you can express the expected evolution in this environment very simply just as a function of the additive genetic variance in the extreme environment. And in this case, we assume the genetic variants are the same in all environments. And then the expected directional selection on the expressed trait value, whether it is in the common or the extreme environment, you could have the selection on the expressed trait value, and this causes a correlated response of the trait that is not expressed in the other environmental value. And then depends also of the odds ratio of the rare over the common environment and the genetic correlation of the trait value across environments. You could measure a trait at 25 degrees Celsius. You can measure the same traits as 26 degrees Celsius. Will this trait be allowed to differ a lot? Generally not that much, because there are some genetic correlation just because of mechanism how the traits are made. So the point was that the total response to selection is at least as large as the rho, this genetic correlation across environments, and increases with the frequency of the rare environment. The whole point of this was just to say that genetic drift does not necessarily dominate reaction norm evolution in rare or extreme environments. And so we completed this with simulations. So for instance, if you have simple simulations of reaction norm evolution in a fluctuating environment, and so these are the range of environments. Think of temperatures, for instance, and this is their frequency, though. These are the common environments, and these are the rare. Essentially, they never occur. 100 times less than these environments. So what you see in this case, for instance, you have the reaction norm evolving. In the extreme environments, there's lots of variance among different replicates, or at different times, sorry. And the shape is very erratic itself. So this is mostly similar to Gallenborg et al. Described. But as long as you increase, either you increase the genetic correlation across environments. So here it's 0.9. It just means that the trait value at 1 degree Celsius differences have 0.9 genetic correlation. But then the genetic correlation decreases as you take the trait in more different environments, obviously. So then the reaction norms become much less erratic. And then if you just increase the frequency of these rare environments, you say they were rare, but they're encountered every once in a while, even though they're on the sort of just on the extreme part of the range and they're rare, just this is sufficient to basically make the reaction norm track the optimum in most environments. So this was just to say that these arguments about relaxed selection in extreme environments, they really need to be made more explicit about what do you mean by rare and how is it that the reaction norm evolves completely freely in these environments. And another point I'd like to make about novel environments is that climate change does not only alter the mean environments, but also the autocorrelation of the environment and the cross-correlation between different environmental variables. This is very well documented. And this will first influence what's the optimal Q for phenotypic plasticity. So this may lead to the evolution of plasticity that depends on different combination of environmental variables. So we studied this in a paper last year. But here what I want to focus on is more what happened even if you just have one environmental variable that determines the trait, But then suddenly, not only its mean shift, but also its predictability, which you can see that here there's high predictability, large autocorrelation, and then it becomes much more noisy. So you can think the Q becomes more or less informative suddenly. So then in this case, if you combine this with a model of evolution of quantitative genetic model of linear reaction norms, you get a dynamics as was described in this paper by Landy in 2009, where basically you have a first phase where, because you have shifted the mean environment and the mean optimum, the reaction norm evolves to higher plasticity because transiently this allows reaching the optimum faster. And this has been described as, can be described as the Baldwin effect. And then in the second phase, which occurs over a much longer time, plasticity decreases. Actually, it goes back to the level that's determined by the current or the new environmental predictability in the new environment, right? So this two-phase process has been described. Then what you want to know is how this affects the load that's caused by those fluctuations. Because when you have a fluctuating optimum that's fluctuating randomly, then there's a, of course, there's a variance of this mismatch with optimum, and this is affected very much by plasticity. For instance, if you have constant plasticity with a slope alpha, then you can say what's the effective variance of the optimum, or the fluctuations of the optimum for the non-plastic part of the trait, say the trait in a common environment. And this is what it is. So it depends on the variance of the optimum, but it also depends on plasticity and how it relates to the correlation between the environment of development and the environment when selection acts on the trait. So plasticity can either amplify or buffer the fluctuations, and this is actually minimum when plasticity equals this correlation term here. Then when you know this, you can compute the expected demographic load caused by these random deviations from an optimum using quantitative genetic theory, conditional on actual plasticity, on current plasticity. And then when you have evolving plasticity with the trajectory that I showed before, you can use this transient, you can use this load conditional on current plasticity to get the whole trajectory of this stochastic load caused by these fluctuations. So then you can plug this in a population dynamic model to investigate evolutionary rescue caused by changing plasticity in a new environment where predictability has changed. And just one simple point I want to make with this is that so this stochastic load, it causes variance in population growth, and as a result, the population size can span several orders of magnitude. And this in itself contributes a lot to extinction risk. Just if you start different replicates, they will end up in different places. And then in the evolutionary rescue, what happens for evolutionary rescue? If you look at the growth rate at the end of this phase one, where you essentially reach the mean optimum, then what you need to remember from this picture is that the growth rate and then the probability of evolutionary rescue is strongly dependent on this new the predictability of the environment, of the novel environment. And the reason is that if you have low predictability, just because you have plasticity that's transiently increasing during the evolutionary rescue, this will increase the variance among replicates a lot and will cause some population to go extinct. So, yeah, I guess, I don't know how much time I still have. This is just summing up all these points so that the initial plasticity may facilitate persistence if it's adaptive, or persistence in a new environment, but this depends on how extreme the environment was in terms of how rare it was and how discontinuous selection was, which I didn't really expand on here, and on genetic constraints of reaction norm shape. The evolution of plasticity may facilitate population persistence in a new habitat, but plasticity also has a strong effect on this variance of fluctuations. So if an increase in plasticity causes increased variance of these fluctuations, this can be also problematic and needs to be accounted for. So I'd say there's still little empirical or at least experimental investigation of these questions of interaction between stochasticity and the environment, plasticity and evolution and demography. So hopefully one day I can present some results on this. Not today.
I'm afraid afraid we'll have to move straight on. Our third talk is from Tom Van Doren from the University here in Marie Curie. And he is talking on assessing adaptation to climate change in evolving bed hedging traits with phenotypic plasticity and maternal effects.
Okay, thanks Raj and Stu for the invitation to this conference. I've enjoyed it much so far already. So I'm going to talk about adaptation or adaptational lags in bed hedging traits. Now, climate change is going to happen for sure, and all we discuss now is actually how fast it will happen. But for sure, there will be some point where the beach will be in Paris for the entire year. Now, it already is, but only in August. Okay. But then if we see how people are actually assessing whether adaptation to climate change is happening, there are quite some studies that focus on detecting responses at the genotypic level, so actually demonstrating a selection response. And we know from quantitative genetic theory that the direction of selection response is not necessarily in the direction of the optimum, not straight to it. And when there is frequency-dependent selection, you might even move further away from an optimum for selection. So I believe that just testing for selection response is not enough. Fortunately, there already are a number of models which actually try to estimate adaptational lags that I will discuss today. So, and this is like a simple explanation of how this works. So, over time, a population is trying to approach an adapted state. This adapted state can either be constant over time, over the time period, or it can change with a trend, as we expect it to happen when climate change happens. Now, when this adapted state is staying put, then the population average will gradually approach this adapted state. When it's moving, then we expect, because of a balance between the selection gradient, the strength of selection, and the amount of genetic variation available, it has been found that in a number of scenarios, that the population will lag behind the adapted state at a fixed distance. And there's a nice review on models of this style, to which Louise contributed a few papers, and in a paper by Michael Kopp. Now here there's something surprising for me as a life history theoretician, which is that the adapted states in most of these models were integral parameters of these models. They were selection optima that were fed directly in the equations. And that's often not the case. For example, if we look at this textbook model of evolution of an evolving germination probability of seeds in annual plants, in desert annuals usually, then this evolving germination probability it's not put a priori. The adaptation for that is not put a priori in the model. It has to be derived from evolutionary reasoning. And this model is relatively simple. So it all starts from the population dynamics, where we track the number of seeds per unit of area in a seed bank over a year. And every year seeds can either stay in the seed bank, so not germinate with probability 1 minus p, and then survive in the seed bank with probability s, or they can germinate with probability p. And then the important thing happens for this style of modeling, which is that the recruits that they will produce, their contribution to the seed bank in the next year, will be time dependent. And in the models I consider, there's also some density dependent. So the number of recruits per unit of area has a plateau, at least. It's controlled. Okay, so these are the essential ingredients of the model, for which I will try to determine adaptational lags now. And it's an interesting model. It's part of the folklore of our discipline. And Steve Elner, he wrote a very nice chapter on this model called You Bet Your Life. I encourage you all to read this. It's really great fun. What is important is that if we want to reason in evolutionary terms about this life history model, we have to consider the success or failure of mutants with a certain low density in the population. And to determine whether a mutant will be able to invade or not, we have to look at the long-term growth rate of the number of mutants in the seed bank. So we have to calculate the contribution to the seed bank per year, and then average this over a long time period. And in the limit where this time period goes to an infinite number of time steps, we get what we call invasion fitness, which we calculate for a mutant probability of germinating in a population which is doing something different, so a different germination probability. Okay, so what do we have to remember here? Well, if recruitment, for example, varies over time, then adaptation is determined from long-term fitness, when there's density dependence in our population system, we need to consider the success or failure of mutants in established populations to determine adaptation. And we get like game theoretical arguments, tournaments between mutants and residents based on invasion fitness. And this style of evolutionary modeling is called adaptive dynamics. Qualitative conclusions are that there can be a long-term fitness advantage of not always germinating, so spending more than just a winter or a summer in the seed bank. And this happens when recruitment fluctuates enough. And in these evolutionary tournaments for this model, there is a single winner, which is a germination probability that we can call the adaptation, because if it's common in the population, no mutant can displace this genotype from the population. Okay. But all of these simulations, these calculations, they assume that there are no trends in the environment. And during climate change, we will for sure have a trend. It's already there. So then the question becomes, what is the environment that is changing? How will we describe it exactly? And how can we determine adaptation in that case? Well, it starts by seeing the environment as a realization from a stochastic process where the parameters vary over time. So if we say that in this century that the average temperature on the planet will increase by 2.5 degrees, let's say, then we're actually describing a stochastic process where temperature will fluctuate between years, but the trend will be described by a gradually increasing average temperature. And every year a sample is drawn from this stochastic process with the parameters of the process fixed at time-specific values. Now in the model, if we're going to look at adaptation to climate change, I will assume that these parameters change gradually. That's actually what everybody does. Okay, and then to make things concrete, I assume that years can be good or bad. There's a probability that the year is good. In good years, the number of seeds recruited to the seed bank is K for the entire population per unit of area. In a bad year, it's small k. And the survival probability can also change between years. So to describe the environment, we need 4 parameters that change over time. But then for each individual set of parameter values, we can determine what the adaptive state would be. And we can make diagrams like this that represent variation in 4 parameters, so survival here, probability of a good year. And then for each combination of recruitment in a good and a bad year, you can determine an area where it's good to spend more time in the seed bank and another area where it's bad to spend more than one winter or one season in the seed bank. And climate change might make us follow a trajectory through this parameter space where you move from parameter ranges where seed banking is adaptive to where it is not. There's also possibilities possible where you can go from an area where you should not spend more time than necessary in the seed bank to spending more time and back again. There's all sorts of things possible. And we can call this idea that climate change gradually changes the environmental parameters and the adaptation follows along. We can speak of this as continuation. Okay, so here's like a almost silly looking example where we have time over 5 centuries or 500 years changing it, but it's just an artificial example. And this is recruitment in good years, so it becomes worse throughout this period. In bad years, it gradually increases, so the nature of good and bad flips. Survival probability goes gradually down, and the probability of a good year as well. So for each of these parameter values, you can determine for every time-dependent set whether it would be advantageous to spend more than one season in the seed bank or not. So we go from a window where it is advantageous to have a p smaller than 1 to where it is not, to where it is that back again. And then comes the essential calculation here, which is calculating a population trajectory. So I made a model with rapid evolution, clonal organism, and 1% of all offspring individuals are mutants with a new value of p. And that's producing, if we follow these changes in the parameter values and we sample this stochastic process every year, this is a population trajectory that we get. For each set of values, we can also determine the adaptation, so the p's from adaptive dynamics, and that's giving this. So you see it becomes 1 in the time window where I can calculate that analytically. And the difference between the two becomes the adaptation and lag. So that's how you do this for a life history model, where you don't know beforehand what the adaptation should be. please note here that the lag can change in sign. Okay. So you can lag ahead or behind, either way you want to call it, but this can flip back and forth quite a bit during periods of climate change. You can do an approximate calculation where we just draw repeated samples from time-fixed states of the parameters, and then these become the predicted adaptations. And you see we don't reach this boundary here, and that's so usually in this kind of models, if you assume rapid evolution to find the adapted states, you get the mutational pressure that keeps you away from the boundaries. So it's only approximate, so you get slightly different values of the lag. So to understand where the adaptations are, you don't have to look at rapid evolution, but at slow evolution. That's also essential. Okay, I'm going to skip this to save a bit of time. That's the definition of the adaptation lag. So it's the difference between the endpoint of adaptive dynamics and individual genotypic trait values at each time point during climate change. And we can average these differences or calculate them for subsamples if we want. But that's the essential thing. Then I extended the model with plasticity and maternal effects. So then I model an underlying liability, which is a sum of genetic values of a plastic response to a cue from the environment and another response to a cue from mom. And a nonlinear function mapping the liability to a probability. The maternal cue is given to new recruits by their mom. They keep this and respond to it later. Plasticity is a response to the same state of the environment at germination. Okay, here are the results, the same environmental sequence. Here are the population responses. That's not so easy to interpret, but you can note that plasticity evolves to positive liability values when good years are really good, and to negative values when good years have become bad. The adapted states, I didn't determine them in this graph from adaptive dynamics, but from rapid evolution. And you see again that plasticity, for example, is not evolving to the boundary where it's expected to be according to adaptive dynamics. So also here, in multi-trait models with plasticity, we have these mutational pressures. Then I can calculate the lag as an Euclidean distance between the population average and the adapted state. And you see that it's fluctuating over time. So we have a forced dynamical system here with a time-dependent input in a way, and the trajectory is not very obvious. If you look at the lag in 3D space, you see that it can make quite funny moves. So for more complicated models, you can already anticipate that the lag will not really decrease in a very straightforward way. Okay, so these are my conclusions. So plasticity and maternal effects in germination probabilities will evolve as well to accommodate the sequence of environments encountered. You might have noted that the maternal effect is often opposite in sign in response to plasticity. It counterbalances it very often, and I call it the maternal muffler. It's sort of reducing population fluctuations and responses. And what you sometimes see in simulations is that traits evolve in compensation for the lag in other traits. And then the question is that part of our picture of adaptation too? I haven't really figured out. That's something we might discuss tonight or tomorrow. Okay, and then we want to apply this to real-world systems. Philip Ginap has given it a trial already, but he didn't do the life history modeling throughout. He went from results from a life history model and then plugged the results into a quantitative genetic model as Louise would use it. But we plan to do it all the way in the future. Okay, and that's it. Thank you.
Thanks, Tom. We do have time for just a quick question before we move on. Yeah, please.
Is there a way to...
Yeah.
I can shout.
You can come here as well. You can use this one as well. Yeah.
Is there a way to predict analytically what this lag should be? And also, can we know how this relates to a load? Because in model without frequency dependence, we are interested in the lag because this directly determines the load for population growth.
Yeah, I believe that for some life history models, it's possible to predict the lag analytically. But not, of course, not in general, but at least for specific models, it must be feasible. And then there are many definitions of loads lag. There's a John Maynard Spitz paper on the lag load, it's unclear at this point how it relates to these ideas of adaptation, because even in Maynard-Smith's models of lag load, the optima were sort of predetermined and not emerging from the life history calculations. So it remains to be seen how all that matches up together, all the concepts used in these different visions of what happens. Thanks.
Okay, so I'm now delighted to be able to...
But I will eventually. Thank you to the Stu and Raj for inviting me to speak here. So I've sort of broadened my talk somewhat because I've got a little bit more time than I thought I had. So I'm going to really be thinking about the effect of recombination on the rate of adaptive evolution. So really thinking about really thinking about whether organisms can respond to environmental change, what limits their response. Okay, so when we compare two species, a species and their most closely related relative, typically we find that they differ by a few percent. So typically not by very much. So this is Drosophila melanogaster and Drosophila simulans. They're about 10% divergent. But of course, genomes are so large that actually that adds up to a huge amount of nucleotide differences. So just thinking about the sequences that you can align, these two species differ by about 15 million differences. And that doesn't include all the sequences that one species has got and the other hasn't. So a question that, of course, has intrigued evolutionary biologists for a long time, particularly molecular evolutionists, is what proportion of those differences are really just neutral mutations which have been fixed by random genetic drift, and what proportion of those differences are due to adaptive mutations. So we'd like to know the answer to the question about, all the differences we see between two species, but that's still a pretty difficult question because we don't really know what parts of the genome are actually subject to selection. So the vast majority of non-coding DNA, some of it clearly regulates gene expression and so on, but actually finding those sequences is pretty difficult. So we've really focused on the question of what happens in the protein coding complement of the genome. But even within the protein coding complement of the genome, we're looking at a huge amount of difference between these two species. And this is true for pretty much every species that you look at. A species and its most closely related relative will differ from that relative by thousands, if not tens of thousands, if not hundreds of thousands of amino acid differences. So again, that question is really what proportion of those differences are a consequence of positive adaptive evolution and what proportion are just neutral mutations fixed by random genetic drift. So we can potentially get at that question by using an idea really first put forward by Dick Hudson and then sort of developed by Marty Kreitman and John MacDonald. And that's really to combine data from within species and between species. So we're interested in what's going on between species, but we can get an idea about what proportion of the differences we see between species by looking at what's going on within species. And the reason that we can do that is basically because most of the variation that we see within species is dominated by neutral and slightly deleterious mutations. But when we look at the differences between species, then we can get both neutral, slightly deleterious, and advantageous mutations contributing to the differences between species. So the crucial thing in the sort of MacDonald-Kreitman framework is the fact that advantageous mutations can contribute substantially to divergence, but they don't tend to contribute very much to the polymorphism within species unless they're balanced polymorphisms. And I have to say I'm going to just sweep that complication under the carpet. We really know nothing about balanced polymorphism in any species. I'm hoping to do some work on that in the near future. So it's this contrast really between what goes on between and within species that allows you to get some idea about what's going on in terms of adaptive evolution between species. So the idea here really is to use your polymorphism data to infer something about the distribution of fitness effects. So we basically summarize the variation within a species in terms of what's called the site frequency spectrum. So we count the number of positions where you've got a singleton. So we've got 4 Gs here and 1 C. Number of sites where we've got a doubleton and tripleton, etc. So this is basically the allele frequency distribution in your sample of DNA sequences. Turns out that that gives you information about the distribution of fitness effects. So we're going to use this to infer this distribution of fitness effects. Now, there are some complications in doing this. Demography is a big complication because demography also affects the site frequency spectrum. We've got a couple of different ways in which we basically get rid of demography or control for demography and infer the distribution of fitness effects. So what we're doing here now is we're taking this distribution of fitness effects, which we've inferred from the variation within a species, and that allows us to predict how many neutral and slightly deleterious substitutions we expect between species. And if we see more substitutions than we would predict, then we infer adaptive evolution, and not only that, but we can actually estimate how much adaptive evolution there's been. So what I've given you is a sort of run through on the Air, Walker and Keatley method, which is basically a McDonald-type approach to estimate the number of adaptive substitutions, correcting for slightly deleterious mutations by estimating this distribution of fitness effects. Now, one thing I haven't emphasized is that when you're doing this, you need a source of neutral sites. You need this sort of neutral standard relative to, so you can judge everything relative to those neutral sites. For most of what I will be talking about, we're going to assume that synonymous sites are neutral. And then we're going to be thinking about, well, what's going on at the non-synonymous sites, so the changes in the protein coding sequence which can change an amino acid. Okay, so I'll just start you off with an example. So this is actually some work from some time ago. So this is comparing Drosophila melanogaster and Drosophila simulans, so looking at the amount of substitution, the proportion of adaptive substitutions between these two species, using a data set of just 420 genes. From 12 African Drosophila melanogaster lines. The significance of using the African lines is because this is where they originated. Therefore, the demography is probably rather simpler than what you get in European lines. That just simplifies things somewhat. So we apply our method to the species, and we get the answer, which is that about 50% of all amino acid substitutions are inferred to have been fixed by positive adaptive evolution. And the confidence intervals in this are reasonably tight, between about 40% and 60%. If you now, now we've got whole genome sequences. If you just do a whole genome analysis, you get very much the same estimate. In fact, you got very much the same estimate when we did this in 2002 with just 35 genes, just showing you that you can use a statistical sample to infer something about what's going on. Okay. So this sort of procedure of estimating the proportion of adaptive substitutions has been done by us and other groups in a wide variety of species now. So we've probably got more than 60 or 70 estimates. And I'm just giving you a flavor for what those estimates are. The upper limit seems to be sort of the 50-60%. There are some estimates which are a little bit higher than this, so things like E. coli, around 60%. This is a relative of the house mouse, around 60%. Drosophila species often come in around 50%. But there are some species actually for which we have almost essentially no evidence of adaptive evolution. Clearly they have adapted, but as far as we can make out, it hasn't been in their protein coding sequences. So things like ZMAs, and this should be actually Saccharomyces paradoxus, the wild relative of Saccharomyces cerevisiae, both coming in as essentially zero. And humans come out as being really quite low as well. Around 10% of amino acid substitutions between humans and chimpanzees have been inferred to be due to adaptive evolution. So we got this variation, and of course we're intrigued to know, and this is statistically significant, this variation. So of course the big question is why we see this variation. Why do some species appear to have higher rates of adaptive evolution than other species? And there are lots of different explanations possible. The first is that actually the statistic I've been using so far is this proportion of adaptives, or proportion of substitutions which are adaptive. So a species might have a high proportion either because it has a high rate of adaptive evolution or a low rate of neutral substitution. So if 50% of the substitutions are due to adaptive evolution, then 50% of them are also due to neutral evolution. So it might be that actually this, some of this variation we're seeing is just variation in the rate of effectively neutral substitution. Well, we can actually basically get rid of those effectively neutral and have a statistic which doesn't depend upon them, and you still see substantial, significant variation in the rate of adaptive evolution. It can also be things due to methodological biases. The methods are not perfect by any means. Not very much more I can say about that. But there's also a third possibility is, of course, ecology. So organisms do differ in terms of their environments and how rapidly those environments change. And I'm sure that this is one of the things going on, that some organisms have more rapidly changing environments than others. But one of the big challenges is actually working out for an organism how rapidly does its environment change. And I think that's a really big and difficult thing to do. There's also been a lot of debate about whether effective population size is important, so whether organisms with larger effective population sizes evolve or adapt faster than those with small. You might expect that under very simple models of evolution. So if the rate of adaptive evolution is limited by the supply of mutations, so an organism is sitting there, its environment changes, and then it has to wait for those advantageous mutations to occur. If that's the case, then species with large population sizes will tend to adapt faster than those with small population sizes, basically because there's many more individuals there for those mutations to occur in. As I say, there's some evidence that there is an effect of population size, but there's also some evidence that there isn't. So I think the jury's out on that one. So what I really want to concentrate on today is actually Hill-Robertson interference. So what is Hill-Robertson interference? This is where selective mutations essentially interfere with each other's dynamics. So in terms of advantageous mutations, what Hill-Robertson interference does is it reduces the probability of advantageous mutations being fixed. So I'll just give you two sort of cartoonish examples in which this occurs. So let's imagine we've got some chromosomes here. These are these bars, and we've got these blue dots. These are deleterious mutations. So every population has deleterious mutations constantly coming into them. So there's a standing population of deleterious mutations. Then we have an advantageous mutation which occurs. Now, on average, that advantageous mutation will occur on an average chromosome in terms of the number of deleterious mutations it's got. So it's not going to be on one of the best chromosomes. And therefore, this is going to reduce, if it can't get rid of these deleterious mutations by recombination, it's going to reduce its probability of fixation. So if there's no recombination, you hopefully can see that this advantageous mutation has a limited chance of being fixed. If these two deleterious mutations, for example, were more deleterious than this is advantageous, then this just gets immediately lost from the population. It gets selected out. Another sort of Hill-Robertson interference, these are actually two sides of the same coin, is of course the sort of classic Fisher-Muller explanation for the evolution of sex. So let's imagine we've got two advantageous mutations at two loci with no recombination between them. Those two advantageous mutations are competing against each other to fix. If they can't recombine onto the same chromosome, one of them must ultimately be lost from the population. So again, this sort of Hill-Robertson interference is reducing the probability of fixation. Now, Hill-Robertson interference is going to depend upon the recombination rate and also things like the density of selected sites and the number of genes or selected sites in the vicinity of a locus that you're interested in. So it may be that part of the reason that we're seeing variation in the rate of adaptive evolution is actually there is variation in the level of Hill-Robertson interference between species. Now, I'm actually not going to give you any evidence that that's the case. What I'm just going to try and demonstrate to you is that in at least one organism, Hill-Robertson interference seems to have a major impact on the rate of adaptive evolution. So that organism is Drosophila. What else? So we're actually just going to compare different regions of the Drosophila genome and see whether we can pick up the signal of Hill-Robertson interference affecting the rate of adaptive evolution. So we've got a data set where we've got just over 6,000 Drosophila genes where we're taking polymorphism data from Drosophila melanogaster from something called the DGRP. This is the Drosophila Genome Resource Project. And we're looking at the divergence between melanogaster and Yakuba. We've chosen Yakuba rather than simulans because there are some technical difficulties with using simulans. It's rather too closely related to melanogaster you can get some complications with very closely related species. We've got some recombination rate estimates. So we're going to be looking at the rate of adaptive evolution as a function of recombination rate. So we are predicting that genes in regions of the genome with low rates of recombination will have more Hill-Robertson interference than genes in regions with high rates of recombination. We're also going to predict that actually genes in regions of the genome with high gene density will also suffer more Hill-Robertson interference. Then we're going to measure the rate of adaptive evolution rather than looking at the proportion of substitutions which are driven by positive adaptive evolution. We're just going to look at the rate of adaptive amino acid substitution, something we've called Ka+. And our neutral standard are synonymous changes at fourfold degenerate sites. And we're going to be using that method to infer the rate of adaptive evolution. Now, one of the problems with using this method is it needs a lot of data. So you need a lot of data to get a sort of sensible estimate or a robust estimate. And that means we have to group genes together. And we've done that basically on the basis of recombination rate. So we just basically rank all of our genes by their recombination rate, and then we take the bottom group of 136 genes as one group, and then next group is the next up, lot of 136 genes, etc. So for each group of genes, we're basically going to estimate that average recombination rate and their rate of adaptive evolution. So this is what you see. So we've got recombination rate along the x-axis going from no recombination to basically some recombination, and we've got our measure of adaptive evolution basically going from low to high. This is actually zero adaptive evolution. Occasionally the method throws up negative estimates, which of course are meaningless. This just means you have to allow negative estimates to statistically, otherwise you get very biased estimates. These basically mean that you have no evidence of adaptive evolution. So actually you can see this very nice curvy linear relationship here between the rate of adaptive evolution and the recombination rate. So our inference here is that what's happening is that when you get sort of out into this part of the curve here, you've actually got enough recombination to break up any associations between advantageous mutations and other advantageous mutations and deleterious mutations. So essentially there's no Hill-Robertson interference out in this part of the curve. But actually, when you get limited recombination, then you're starting to get your advantageous mutations tangled up with each other and with deleterious mutations, and that's reducing the rate of adaptive evolution to the point when, in fact, you've got no recombination, you've actually got no adaptive evolution. So if you take the mean of all the points which are right down here with zero recombination, you basically have an estimate of no adaptive evolution in these genes. So one of the things you might want to do is try and estimate, well, how many adaptive substitutions or potentially adaptive substitutions are being lost because of Hill-Robertson interference. So we can take this asymptote as basically our estimate of the rate of adaptive substitution that you would get if you had no Hill-Robertson interference. And then this depression here is basically the effect of Hill-Robertson interference. These are adaptive mutations which are coming into the population and are being lost because they're getting tangled up through Hill-Robertson interference. So basically, we're going to sort of draw a line through this asymptote here, and then basically look at the difference between this line here and the sort of line of best fit through these points here. And when you do that, basically what you infer is that if you had no Hill-Robertson interference, so if you basically boosted all of these points up to this line here, you would have in this data set 66,000 amino acids, adaptive amino acid substitutions. But actually the difference here is actually 15,000. So 15,000 of those have been lost because of Hill-Robertson interference. So actually we're finding that about 27% of all adaptive substitutions that would have gone to fixation if there was no Hill-Robertson interference, 27% of those are basically being lost because of Hill-Robertson interference. So that's really quite a substantial effect. I mentioned also earlier that we expect Hill-Robertson interference to depend upon gene density. So this is just gene density going from low gene density to high. This is basically looking at the number of coding sites in a 100 kb window centered on the gene of interest. This is, again, our measure of adaptive evolution. You can see there's very nice negative correlation here. So we have some evidence that Hill-Robertson interference can reduce the rate of adaptive evolution in at least one species, and so it is possible that it's causing some variation in the rate of adaptive evolution between species, but I have no evidence of that as of yet. That's something that we're working on. But Hill-Robertson interference is going to depend upon the rate of adaptive evolution, the distribution of fitness effects of deleterious mutations. It's going to depend upon genome size, gene density, recombination rate, and effective population size. So there are lots of factors which affect the level of Hill-Robertson interference, so it is possible that some of those are varying and actually altering the overall rate of adaptive evolution between species. So now I'm going to come back to the sort of original idea of my talk, or original focus of my talk, which was mitochondrial DNA. So when we're looking at Drosophila, we're really basically seeing no adaptive evolution when there's no recombination. So mitochondrial DNA is, of course, a classically non-recombining molecule. So you might ask, well, what's going on in mitochondrial DNA? Are mitochondrial DNA also down here at zero? Or do we actually see some evidence of some evidence of adaptive evolution in mitochondrial DNA? So to do this, what we did was take a rather old data set of 1,000 animal species for which we've got multiple sequences from within a species and at least one outgroup. And I regret actually taking this old data set. We should have really compiled a new data set because there's stacks more data out there. But we didn't. We were a bit short of time for various reasons. We restricted it so DS at a level of synonymous divergence has to be at least 10%, because if your outgroup's very close to your in-group, you can have problems. And we want to avoid them being too saturated, so we've restricted it to data sets which don't have very high DS values. We've restricted to data sets in which we have at least 12 sequences. If we have more than 12 sequences, then we've basically downsampled to 12 sequences. We have to do that because basically what we're going to do is combine data across species, and this leaves us with 372 animal species, for which we've got multiple sequences from within a species and at least one outgroup. And we're just using one outgroup. So one of the problems, again, of applying this sort of methodology for inferring the rate of adaptive evolution to mitochondrial DNA is basically twofold. First of all, you don't have very much data per species, so you can't apply the methodology to each species in turn. And secondly, because you've got no recombination, the site frequency spectrum tends to be very, very erratic, because basically all sites share the same genealogy. So you can get these very odd-looking site frequency spectra. So we were realized that actually what we could do is basically just take all species and lump them together. Now, to do that, really what you want to do is actually lump them together in a way that weights each species equally. Otherwise, a species with huge amounts of polymorphism data you'll get the answer from that one species. So we've got a methodology which basically takes the site frequency spectra from each species and the divergence data from each species and actually combines them in a way that weights each species equally. So when you do that, you get a combined site frequency spectrum and a combined set of divergence data. And what I've shown you here is basically that composite site frequency spectrum, but actually I've just shown it as a ratio. So this is the number of non-synonymous polymorphisms over synonymous polymorphisms for each of the frequency classes. So these are singletons, doubletons, tripletons, etc. We've only got six frequency categories because we can't orientate our SNPs. Our outgroups are too divergent typically. So we don't orientate SNPs, so the site frequency spectrum is what we call folded. And you can see it sort of takes on the form that you would expect if some of these non-synonymous mutations are slightly deleterious. They tend to segregate at lower frequencies than the synonymous variants. But what our method is effectively doing is then fitting a distribution of fitness effects to this sort of ratio of Pn over Ps, and then using that to infer, well, what do we expect in terms of divergence? And if we see more non-synonymous divergence than we'd expect, then we infer adaptive evolution. And when you do the maths, then you come up, or the estimation, you come up with an estimate around 26% of all substitutions in mitochondrial DNA have actually been driven by positive adaptive evolution. Confidence intervals, unfortunately, in this are quite large, 6% to 45%. This is basically because although you've got 372 species, actually you're weighting those equally, and some of those species have very little data. So again, I regret that we didn't go out and actually compile a much bigger data set and try and resolve these confidence intervals much better. So you might be thinking, well, what's going on here? So, from the Drosophila data, we were predicting that actually if you have no recombination, basically you have no adaptive evolution, but we seem to have got some adaptive evolution in mitochondrial DNA, the molecule which is classically non-recombining. Well, you can resolve the difference between these two sort of data sets by realizing that actually, although mitochondrial DNA has 26% of its substitutions are adaptive, actually it has a very low rate of substitution. So actually, when you plot it on this graph, it falls very much in the sort of pattern that you would expect from Drosophila. So although 26% of the substitutions are adaptive, actually it undergoes so little substitution that in fact it fits very much in this sort of overall pattern. Okay, so that just leaves me to sum up. So what I've shown you is that Hill-Robertson interference can substantially impede the rate of adaptive evolution, and maybe this is one of the reasons that rates of adaptive evolution differ between species. Nevertheless, we do see that you can get some level of adaptive evolution in something like mitochondrial DNA, even though it's non-recombining. So that just leaves me to thank a couple of people, the two people who really did most of the work, David Castellano and Jenny James, who are both PhD students. Actually, David wasn't my PhD student, he was somebody else's, but I stole him for a while. And Jenny James is a PhD student with me, and then a few other people have helped along the way. Thank you very much.
Thanks very much, Adam. We've got time for a couple of questions.
Yeah.
So, in recent years, there has been quite a bit of work on. In recent years, there's been quite a bit of work on what's come to be called conditional mutations, mutations with conditional effects that may be deleterious when they first arise, but they may then turn beneficial, perhaps in a changing environment. We don't know. So there are, for example, a lot of transfer RNA mutations that are deleterious in humans. They are associated with disease, but they are fixed in primates. So when you calculate an overall fraction of positively selected mutations, how can we think about these conditional mutations?
Yeah, so we're always thinking about the net effect, the overall net effect. So when we say something's deleterious, we're saying that on average it's deleterious, or its dynamics sort of average are basically deleterious or advantageous. So I think although those effects sort of come out in the wash. Yeah, thanks. Yeah.
Any other questions?
So are you assuming with your comparisons that the recombination rates for particular groups of genes stay the same over long periods of time?
Yeah, we don't know that. Yeah, so we're just taking what's occurring in Drosophila melanogaster. So, in primates, of course, we know that recombination rates on a very fine scale evolve very fast. They don't evolve very fast on a sort of large scale. I'm not sure that we know so much about what goes on in Drosophila. Just trying to think if anybody's... There must be a recombination map for simulans. Yeah, but it's a good point, yeah.
If you compared across closely related species,
Yeah.
you can see how quickly it changes.
Yeah, indeed, yeah. So it's getting that recombination rate map data. So if it does change, that would really sort of come out as noise. Then we're having an imperfect view on, what's sort of... So if it really did turn out that it was changing quite rapidly, and we're just taking melanogaster's recombination rate estimate, then we're actually underestimating the effect of Hill-Robertson interference. Yeah.
So if you just looked at very recent genes, you might get a better fit. Recent ones of a very recent history than very ancient.
Yes, although, yeah, I think it's still really what's happening with the recombination rate. But yeah, no, it's a good point. And we must have, at least for simulants, we must have a recombination rate map. Yeah.
Any other questions? In which case, I have a quick one.
Oh, yeah.
So when you're implementing your method, you're fitting a distribution to the site frequency spectrum.
Yeah.
So how much power is there in your data set for discriminating between different functional forms, and what would be the implications for your estimates of the proportion of adaptive evolution?
It's a very good question. We haven't explored it very much. Other groups have. So in the human case, where you really, these days, have huge amounts of data, there's a paper that I think is about to come out in genetics where they're looking at thousands of humans and they've got, huge power. Actually, the gamma distribution does, which is the one we fit, actually does a very good job. Yeah, there are in some data sets you do favor another distribution, but actually the gamma distribution pretty much looks identical. And the other thing that comes out of that study is that really it seems that so long as you can fit the site frequency spectrum reasonably well, then you get a reasonably good estimate. So even though another model might be favored, if actually say the simple gamma is favored, if it's fitting the site frequency spectrum reasonably well, then actually you get a reasonably good estimate. But it's something I'd like to explore because I think, there's a general question about how much information it really does contain the site frequency spectrum. Now we've got 60,000 human exomes, so in a sense we've got almost unlimited data, but whether we've got unlimited power is not clear. Yeah.
Okay, thank you. I think in that case we will move on to our final speaker, John Bridle from the University of Bristol. So I'll leave John to introduce this talk.
They postdocs, PhD students. Yeah, it's going to go crazy.
Until the slide changes, this seems like an ideal time for a plug. So seeing as it's a session on adaptation to the environment, I'm not sure if there's anybody in the room who doesn't have a PhD yet, but if there is, and you're interested in rapid adaptation and things like that, Liverpool is part of a NERC DTP with York and Sheffield. And if you haven't looked already, there's a whole bunch of fantastic PhD projects advertised on the NERC ACCE website. So I'd encourage you or anybody else you know who's interested in adaptation, who's looking for a PhD, to have a look.
And if I can add to that, if you're interested, if you're if you're a postdoc or PhD student finishing who's interested in a postdoc, there's one available in my lab in Bristol, starting next year, testing some of the very things that Luis was talking about earlier, and it involves collaborating with Luis as well, and that's looking at the evolution of plasticity in plants up and down Mount Etna in Sicily. So it's advertised on EVOLUTIONARY if you're interested. I've just tried it. Are we up to? Should it have started by now?
Yes.
There we go. Okay. Well, thanks very much for the invitation to speak. It's great to be here. I'm going to be returning, I guess, back to evolutionary ecology in a sense, away from explicit population genetics. I'll be talking about how the way that organisms evolve, particularly in terms of maternal behaviour, can affect the way that they experience the environment, the way they perceive the environment. And that can be in terms of the biotic interactions that they experience, that they, particularly in this case through host plant choice, the types of plants that their larvae are going to be growing on are affected by the choices that mothers like this beautiful animal here, brown algae butterfly, the kind of decisions that they make will affect the future of the following generations. So we're going to talk about the way that the sensitivity of organisms to climate change can be affected by evolution of their behaviour, not only in terms of the host plants, but also in terms of the microclimates that those host plants might harbour. This is work that was done by James, mostly, who's a PhD student, ex-PhD student of mine who's in the room, Micah de Jong, who was a fellow, and myself. So butterflies are fantastic systems to try and understand historical and contemporary responses to climate change, mainly because we have fantastic records. One of the increasingly small list of things that's good about the UK is that we have incredibly good distributional data available for many species of organisms, particularly butterflies, sometimes from the 18th century onwards, thanks to the efforts of guys like this and fantastic manuscripts written a very long time ago. We know a lot about their adult and larval ecology as well, so it means we can exactly show historically how they've tracked their distributions in response to changes in the environment, so how well they've matched where their niche is to where their distributions actually are. So we can see the changes in the host plants and changes in temperature, changes in soil type, and how that matches distributions. And one of the main observations that have been made by the work of ecologists like Chris Thomas is that there seems to be a very poor correspondence between these range shifts depending on how specialised organisms are, and in particular how patchy their environments are. So if they're more specialised on particular types of biotic interaction, they'll have much more patchy environments because there'll be limits to where their particular host plants or their particular soil types or the particular prey items they use or parasitoids that limit their range may be found. So at many different length scales, when we look in the British Isles, we find very patchy distributions of species which seem to reflect the biotic interactions that they have, the specialised biotic interactions that they have. And that's been characterised as the problem of the specialist, which is the fact that most species in Britain of butterflies that have very specialised biotic interactions have failed to track the changes in their thermal niche that have been caused by climate change. So there might be habitats up here that would be suitable for these organisms, but haven't colonised those areas because their host plants aren't found there. So this is the kind of problem with the specialist. About 75% of butterflies in the UK that are specialists are diminishing in their, or remaining static in their ranges, despite the fact their thermal niches are shifting northwards. So what we tend to do in biology is we tend to take exceptions to these rules and see what's going on in those exceptional cases. And the UK brown argus is an exceptional case in the sense that it's defined as a habitat specialist, at least for most of its range, and yet it's had a rapid expansion in the last 30 years or so northwards. So these black circles here indicate the new areas of the range, the new parts of England which are inhabited by this butterfly that weren't in 1975. And that seems to have been associated with a shift in host plant use. So in these new parts of the range here, this organism seems to mostly use these type of open disturbed habitats, or often post-industrial habitats here, where they're using a host plant, geranium molly mostly, which is from the geranium family of plants. Whereas in the old established part of the range, for most of their area, they're using these chalk downland plants here, Helianthus nummularium, or the rock rose, which is a Cystaceous species, a very different family of plant. And it's a perennial that seems to have this very warm microclimate, a very stable microclimate. And it's found in great abundance on very steep chalk downland sites. In some parts of their range, the very coastal dunes, which tend to be locally very warm, they will be using these Germinaceous species, often different types of Germinaceous species, but that won't account for most of the distributional range of this species. So it seems like this shift in their distribution of the specialist has been associated with the change in host plant use by the mothers and by the larvae, of course. The question that we wanted to ask was basically, well, has evolutionary change been necessary for this range shift? Does it seem that evolution is actually limiting the ability of populations to track where their ecological niche is in space because of this problem of patchiness or the problem of specialisation? So the first thing we did was what seemed like now very primitive forms of molecular techniques. We used AFLPs and the genomic test to see if there was evidence for adaptation between the recently colonised and long-established parts of the range, so in the colonisation process. And there's a nice paper by James you can read if you're interested, and the answer is yes. It seems from the basis of molecular data there has been selective sweeps associated with this expansion. But as Raj was saying right at the beginning of this session, it's very difficult unless you're actually in the field measuring what the phenotypes are actually doing. It's very hard to know what you can infer from molecular data, even when you have actual SNP data and genetic data that's tied down to a particular locus that does something. It's actually hard to know how it's actually affecting what organisms are doing in the field. So what I'll talk about mostly today, or exclusively today, is what we've been doing in terms of field-based assays of female preference in field conditions. So I'll talk about two things we're doing. There's three main things we've been doing. The first is we've been looking at laying rates of free-flying mothers. So we just basically put out host plants, which have been raised in pot plants from seed. We put them out in 16 quartets at various sites across the range. And then we come back after 14 days and count the eggs that have been laid on each different type of species, different type of plant. And we use that as an estimate of the site-level preference of all of the females. We don't know who's laid what where, but we just got a kind of overall average of what the females are doing at that site. The other thing we can do is we can do move females onto cages, under cages in the actual different sites in the field, look at what they do when they're exposed to these conditions, that single females are exposed to those different plants, and see what they're doing, but I won't talk about that data today. The last thing we can do is actually do transplant experiments where we move individual females and transplant them between sites and see what they do when they're faced with novel hosts or hosts in different places. We're just talking about these data first of all. So as I say, we put out these plants and assay what the females do by counting the number of eggs that are laid on each different type of host plant. And these are the kind of data that you get. Because they're ecological, they look kind of messy. These are 95% confidence intervals based on bootstrapping. But the first thing to notice here is this is just confining ourselves to just the established sites, so the old parts of the range. And we're looking at sites which are on sand dunes, so these old sort of southern sites on beaches here where they are using geranium's dominated. And these ones here, which are mostly rock rose dominated sites, so these are the old established chalk downland sites. And the first thing to notice is there's lots of variation among sites in what they're doing. But if you look on overall at the rock rose sites, or places where rock rose is dominant, which are these ones here, which have the star tells you which host plant is the most dominant locally at that site, you can see that on average they tend to be more commonly using the most common host plant at that particular location. By contrast, if you look at the old established sites that are using geranium, they tend to be using to a large extent the most common plant at that particular location. These data are a bit kind of messed around because there's these kind of dilution effects, because there's lots of variation in the actual rarity of these plants in the actual sites. So if you've got rock rose out at these areas where rock rose is very common, you're very unlikely to get females laying on those places. So you're probably underestimating the rock rose preferences in these particular sites. It's quite hard to get around that. But the overall result from this is that the rock rose preference is much higher, significantly higher in areas where rock rose is abundant than it is in areas where rock rose is not abundant, in the areas that are geranium dominated. So there's evidence both of variation among sites, but also for some evidence for local adaptation in the older part of the range here. By contrast, when you look at the new part of the range, you see a very, very similar profile across these sites. And you see actually no correspondence at all between what the most common local plant is. Sometimes you have plants which are completely absent, which are most common in that area there, but they use a completely different host plant at that particular site. So there's no correspondence in the new part of the range between what the most abundant local plant is, what the host preference profile is, and it seems to be all fairly consistent in this new recently expanded part of the range. So it seems from these data that in the old part of the range there's evidence for local adaptation, it's matching your preference profiles with what the abundances actually are. Whereas in the recently decolonised part of the range, it seems that the profiles are fairly similar regardless of what the abundance is. This seems to suggest there's been this sweep or this rapid adaptation of a few forms which all seem to use the same type of preference profile, and a kind of loss of local adaptation associated with range expansion. We can test that a bit more by looking at some statistics, but to sort of see if we are seeing more variation among sites in the old part of the range. You can see, if you look, these are all the old established sites here, and this is the among versus within site variance based on the number of eggs laid, and we can see there's highly significant differences among sites relative to within sites in the old part of the range. That's not observed when we look at just the recently colonised sites. Okay, when you compare that, as well as another way of looking at this, you can look at female choice among plant quartets, and you can show that also the amount of variance among sites is much higher in the long-established sites than the newly colonised sites. So there's a consistent reduction in the amount of variance within versus among sites. The final thing I want to talk about is the second experiment, which is the acid test, if you like. We actually do reciprocal transplants of individual females across the range. So we do about 300 individuals have been moved from areas in the south up to areas in the north, and we use these old shopping baskets for doing that. They're fantastic for doing reciprocal transplants because the kids can jump up and down them and not damage them. We also can confuse supermarket assistants when you try and buy them, because trying to buy an empty basket is quite a strange thing to do in a supermarket. Of course, it's a kind of crazy existential confusion amongst them, but that's another beautiful thing to use when you're doing transplants. So we can test to see what individual females are doing when they're faced with a different host plant and you move them by reciprocal transplants. And the results here are really fairly straightforward, surprisingly consistent. The main result is when you move individuals from the south, so when you move individuals from the newly colonised part of the range back to their ancestral homes, if you like, back to the south, you find they don't lay any eggs. So it seems like what's happened is actually they've lost the ability to use rock crows as associated with this expansion. Now that's in contrast to when you move the rock crows, individuals from rock crow sites north onto drain, and they seem to be able to use both fairly happily. So we see a strong reduction in fitness when you move them back south to when you move them from north to south. So there's been a loss in their ability to use their ancestral host. So just to summarise, what we think is happening here is we've got this climate-driven range shift that's associated with the shift onto what's basically a very locally rare plant. Geranium is actually locally very rare, but it's very widespread. The organism is not seeing the environment in a patchy way anymore. It's actually able to cross these areas where there's no rock crows around by using actually a very locally, fairly rare plant, but it's found in many sites. It's a very rural kind of plant. And it now specialises in a single host that has allowed it to spread into these newly thermally suitable habitats. And I think when you get these kind of rapid range shifts that are associated with moving to new thermal environments, changing these biotic interactions, they can cause, as the transplants showed, a significant loss of local adaptation, so a loss of the variation that in the future they might depend on for more adaptation. So I think I'll stop there. Thanks very much.
Thank you very much, John. Any questions for John? No, let's do that. Yes.
John, so the fact that they lose the ability to use the original host would sort of suggest there's some kind of cost of being plastic. But as you know, costs of plasticity are rarely ever found. They don't seem to exist generally.
Yeah.
Have you got any thoughts on why they would lose that ability?
No. I mean, I think you could turn the question around and say, why do they have the ability to use these for two very distinct hosts phylogenetically in the old part of the range? And I think that might come down to this, in good years, it might be okay that maybe this is a bet hedging strategy that they employ. where in the old part of the range they'll use geranium in warm years. They'll always lay on geranium when it's there, because in warm years that really pays off. But in bad years, you want to go on rock crows. But I think maybe in the new parts of the range where geranium's growing, it's basically they're growing maybe particularly warm microclimates. But why there's a trade-off, why there's a cost of maintaining both types of host preference, I don't know. I mean, they are very phylogenetically very distinct plants, so there must be some cost associated with being a larvae that's resistant to the defences of that plant as well as another plant. So maybe that shows there is a cost of using both. It'd be a nice thing to study in more detail.
Okay, thanks.
Just following on from that, is there a role for genetic architecture here? So could there be a positive genetic correlation, for example, between the ability to tolerate a warm climate or to disperse northwards and host plant usage?
Well, we do know actually that some experiments that Chris Thomas's student has done has shown that the movement through geranium habitats is much faster than through rock host habitats. They seem to search a lot more. Because they're much more widely distributed, so they're much rarer. So they have to travel greater distances before they lay. But they tend to be less choosy than when they lay on rock crows. So rock crows vary more in quality, but they have to fly further distances to get to geranium. So I think their average movement in geranium habitats is greater. And we actually see an increase in their dispersal morphology associated with range expansion as well, which I didn't talk about.
So it's not impossible then?
No, I think it's probably very likely, yeah. Thanks.
So, John, another possibility maybe is that there is some kind of genetic polymorphism for host plant preference, and that it's only the geranium-laying ones that have migrated north. Is that?
Yes, actually, that could well be the case. There is actually a sweep of those genotypes, and that's still an evolutionary shift.
Well, yeah, no, true. This is slightly interpretation.
Yeah, I think it's a selective sweep of either of those alleles. Maybe it could just be a few genotypes that spread from one particular site. And the genomics that we're using is going to tell us in the future whether that's come from multiple sites or just at one place. But it seems like they can use both types of host plant throughout the old part of the range, not just at one site. Yeah, it could well be. We've got the next stage of the project, we've got a lot of data from individual females being reared in the common garden environment. So not really in the common garden, tested in the common garden environment in the fields to look at host preference of individuals, and we genotype those about 20,000 SNPs, so we have to see if there's what the genetic architecture of the host preference. Sometimes it can be just a few genes.
Great, thanks. I think I'd also like to ask, we had several speakers who there wasn't a chance or time to ask them questions. So before we thank everyone, I'd like to invite any other questions for any of the speakers you've heard here, or indeed we can meet afterwards and kind of discuss later. Are there any other questions from this session? I know I have a few, but I think I'll wait till last until we're outside. But in that case, I'd just like for us to thank all of the speakers. It's been a fantastic session. Thank you.